Alright, Chapter 8, Mitosis and Meiosis. Mitosis is cell division, it's actually identical division of that particular cell. So it produces two identical daughter cells compared to the parent cell. So there's this one cell, right? Oh, sorry, I'm trying to draw it and I couldn't draw. So the cell will undergo a cell division and form basically two identical cells, okay? So this cell disappears basically. It'll end up, that one cell divides like fission, like we saw earlier on. But fission is the entire organism, right? This is just one cell. This is for humans, for plants, all organisms undergo mitosis. Yeah. Okay, so one of the functions is to replace any dead or damaged cells, as shown over here. So if it's like a damaged cell or a dead cell and we need to replace it, it will have to undergo cellular division or mitosis. Also, to grow, right? To be able to have us to grow to our height or even like muscle mass, and etc., we have to undergo mitosis to grow in that sense. And finally, asexual reproduction, which we just talked about, how through asexual reproductions, they'll have to undergo mitosis to develop that particular limb, or just the entire organism itself, or the amoeba itself just divides asexually through mitosis. Meiosis, on the other hand, is the cell division that occurs to form gametes. The parent cell itself is what kind of cell? How many chromosomes? The parents? 23 pairs, right? So a total of 46 if we're talking about humans, right? For other organisms, it's a little different. They have different numbers of chromosomes. And then our gametes, however, will be how many chromosomes? 23, yes. This is 23 pairs, which equals 46 chromosomes. This is for humans, that is. For humans. Okay, and we talked about how having 23 chromosomes is being haploid, right? While being a parent is diploid, so 2N. And the gametes, once again, for males, it's the sperm, and for females, the egg. DNA is our genetic material. It helps us form our proteins. And we'll talk about that when we end up in a week or two, maybe even next week. Chromatin is DNA with some proteins called histones. These proteins help bind the DNA. And chromatin itself is the diffused form of this protein-DNA interaction. And usually you'll see this chromatin prior to cellular division when it's growing and before actually dividing. When it wants to divide, it wants to condense itself into chromosomes. The reason why is because think about if it's really stringy and all about, it's going to be really hard to divide that, right? If you're dividing the cell, it's going to be really messy and trying to pull a really tangled up string away from each other. However, if it's wound up in a condensed form, it's more easily manageable. You could end up taking that wrapped up DNA as a chromosome here really easy to one side of the cell or another. So this is a condensed form of DNA, and within it they have a sister chromatid. This sister chromatid is just this one side of the chromosome. And since it's undergo replication, this other sister chromatid is identical DNA-wise, it's identical to each other. So the sister chromatid over here, that I'm going to color in red here, is genetically identical with this chromatid, sister chromatid here. And they're fused together here at the centromere. This is the centromere. That's where they are linked together.
It's your sister and then it's your daughter.
Yeah.
Wait, what?
We'll get into that later as to what we're talking about. So DNA, like we said, wraps around the histones, right? The proteins. When it wraps around the histones, it becomes this thing called beads on a string because it looks like beads on a string. Just one of these is a nucleosome. So one nucleosome contains like about 4 histones with DNA wrapped around it. When the beads on the string starts to coil even more so, it becomes the condensed form eventually into the chromosomes itself. It keeps on condensing itself more and more to form this chromosome. And what's it called when it's loose again? Chromatin. Okay. Chromatid in itself is just this one side of the chromosome, remember? Yes. So this is a sister chromatid here on this side. This is another sister chromatid on this side. So chromatin is the loose form of DNA. Chromatid is part of the chromosome. And chromosome is the condensed form of DNA.
Say that one more time. Chromatin is a loose form of DNA. Chromatid is
part of a chromosome. There's 2 sister chromatids to make up a chromosome. And chromosome is the condensed form of DNA. The cell cycle consists of the entire period of it forming and also when it's dividing as well. So it consists of interphase. Interphase is this period of time where the cell functions normally. It's growing in this process and undergoes DNA replication. And most of its time, as you can see, it's spent in this stage here, around 90% of its time. There's 3 parts to this interphase. There's the G1 phase, the S phase, and the G2 phase here. What's G6? Is it a song? Oh, okay. Mitosis, on the other hand, is also part of the cell cycle. One important thing is that interphase is actually not part of mitosis, right? It's actually the thing that occurs after or before mitosis. However, the cell cycle consists of both interphase and mitosis. Yes, mitosis, that's how cancer spread is through undergoing unregulated mitosis. So mitosis is when the actual cell is dividing, and it consists of all these different stages: prophase, metaphase, anaphase, and telophase. Cytokinesis is a process that occurs towards the end of mitosis where the cell begins to pinch off. So this cleavage furrow forms when cytokinesis occurs. And we'll go into more detail about these.
Would you call that like the dividing factor of what's happening in the cells?
You could say it's the process of causing it to physically divide. Cytokinesis. Okay. Interphase. In G1, this is the period, the G stands for gap phase. It's a period of time of growth. So they grow in size and also the number of amounts of organelles inside of it. S phase, the S stands for synthesis. So because it's synthesizing DNA, this is where DNA replication occurs. So we're doubling the amount of DNA at this point in time. G2 phase is also another gap phase, and this is where the cell continues to grow in preparation for cell division. It has to be big enough to be able to divide itself. And once again, what is the form of DNA in this particular case? It's is it in its condensed form or decondensed form? Decondensed form, which is called chromatin. Chromatin. Tin. 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 Like Tin Man tin. Okay. At the end of G2 phase, these centrioles will have these proteins that surround it called centrosomes. And these become key role players when we're talking about mitosis. They begin to form. So now we go into mitosis, right? Mitosis, the first stage is prophase. And what occurs in prophase is a numerous of things. One is that the chromosomes condense, right? So it becomes from chromatin to a chromosome. And remember, the chromosome itself has a sister chromatid on one side and another chromatid on the other side. Exactly. And they are linked together. These chromatids are linked together by this centromere. Okay. And these centrioles that we talked about earlier, that has a lot of protein around it, they start to move towards the pole. And as they move towards the pole, these spindle fibers are made of microtubules. Remember that term? These are those thick proteins. These microtubules extend from the centrosomes so that it could eventually attach to the centromeres of the chromosomes. Finally, the nucleus itself dissipates. So the nucleus has a nuclear membrane around it. For those of you who looked at the online chapter of the cell. So it dissipates, and you'll notice a couple things. Notice, first of all, that the DNA is condensing from this mess here into a little more condensed form over here. The nuclear membrane dissipates, and so there's no more nuclear membrane around it. And we also notice here that the centrioles with the centrosomes around it, the microtubules, they're now going on either side of the cell. Yeah. Metaphase is when the chromosome starts to line on this metaphase plate. The metaphase plate is just the midline. Okay, so they line up over here. The spindle fibers from the centrosomes end up attaching to the centromeres of the chromosome. So on the centromeres in cells, they have these proteins next to it, and these spindle fibers end up attaching to it. The purpose of attaching to it is so that we could go into anaphase, which is the next stage. Anaphase is when we end up splitting that centromere and then causing the sister chromatids to split apart. Once the sister chromatids split, they are now called daughter chromosomes. Okay? So this here is a daughter chromosome. Here is the, that used to be its sister chromatid, but now it's a daughter chromosome. Yeah. Okay. And these daughter chromosomes go to opposite sides of the cell due to the microtubules, the spindle fibers pulling it from the centrosome. At this very end of anaphase, depending on which textbook you look at, some textbooks say that cytokinesis occurs in telophase. Other books say that it's at the end of anaphase. So we're going to say late anaphase, early telophase, that cytokinesis occurs. And cytokinesis begins by pulling the plasma membrane on the inner side of it, pulling it in, causing this cleavage furrow to form. Telophase. Yeah. Okay. So in telophase, it's basically the reverse of prophase. So when I say it's the reverse of prophase, what occurs in prophase, first of all? The chromosomes condense. So in telophase, then, what is it going to happen? The chromosomes decondense. And when it decondense, eventually it become what? Tin. Very good. You got it now. Okay. So the chromosome starts to decondense. Okay. What other things happen in prophase?
Centrioles move to poles.
Okay. Yeah. They don't have to. Yeah. Well, I mean, they don't even move together. They just stay at their current location.
So then does that mean that two nuclei form in both spots?
Yes. Also the nuclear membrane dissipated, right? So in this case scenario, telophase, we're going to reform that nuclear membrane. The nuclear membrane forms again, so we have a nucleus once again. The nucleolus, remember the nucleolus, its function was to make RNA. It reappears. This is within the nucleus itself. Why does it do this? It's basically resetting itself to become an individual cell again. Remember how we decondensed we dissipated the nuclear membrane purposely so that we could end up causing cell division to pull the chromosomes away from each other to form two cells, right? So we're resetting that now. So we're reforming the nucleus. We're going to decondense the DNA to become back into an interphase cell.
So basically it just comes together, mixes, and then separates again?
It doesn't. Okay, so I guess we're missing a point here. So here, originally, here's a cell, right, with its the nucleus, right, with the nuclear membrane around it and stuff like that. And this is what's in here. What is that? It's what is it? Like this is the nucleus, right? The DNA inside is called what? The decondensed form? Chromatin. Yes. Okay, chromatin. Okay, so as we go into prophase and everything, the nuclear membrane will start to dissipate. Okay, the chromatin will become chromosomes now. Okay, so, and then we have also the spindle fibers. I'm not going to draw it in, forming to eventually attach it to it. So these chromosomes will then, in metaphase, then line up in the middle on the metaphase plate. So now we have the chromosomes lining up here. Okay, that's metaphase now. Okay, and sorry, I should draw these more like that, since the chromosomes itself looks more like X's, right? Where, you remember the chromosome, right? They have a centromere here. So this side of the chromosome is a sister chromatid. Yes, and this is another sister chromatid. At this point in time, I'm going to draw this centrosomes with its spindle fibers. The spindle fibers will attach to the centromeres. There's some proteins around the centromere, and it will attach to here on this side and attach to here on that side. Okay, and in anaphase, the centromere here will break. It's going to break so that now the sister chromatids can separate. Once they separate, so I'm going to draw that in here. I'm going to erase that. Now that they separated, okay, they are called what now? Once they're separated, daughter chromosomes. Very good. So these, yeah, you're on it. Okay, so this is a daughter chromosome. Very good. Okay, and now they're pulling these chromosomes apart. Okay, these are, remember, undergoing, we already duplicated the DNA interphase during the S phase. So these identical DNA are getting pulled apart now. Okay, so they're getting pulled apart. Cytokinesis starts to occur. And I'm going to redraw it like this. Oops. And then we had the spindle fibers. I'm going to draw the centrioles and the centrosomes, blue color this guy. Okay? They get pulled apart, right? And now at this point in time, this is anaphase, right? We're going to go into telophase, where we end up reversing the process of prophase. So what's happening here is we're reforming the nuclear membrane. We're going to end up decondensing the chromosomes, the daughter chromosomes, into chromatin again. And eventually, cytokinesis will complete itself, and now we have how many cells? Two complete cells. And notice how this looks identical or almost identical compared to the first thing that we started off with. Yeah? So does that make more sense as to why we're doing the reverse of prophase and telophase? We're doing the reverse just to form two normal cells again. Okay, cytokinesis will complete forming into the two daughter cells. So I just explained that. It's getting super dark now.
It's time to go home.
Is it? Alright, cytokinesis, we already talked about this, how it begins at the very end of late anaphase or early telophase. And in animal cells, they form this cleavage furrow, and this is due to these microfilaments pulling on the inside of the plasma membrane. And when they pull it from the inside of the plasma membrane, it's going to cause this indentation called this cleavage furrow. Versus plant cells, they do it differently. Since they have cell wall around their cell membrane, they form a cellular plate or cell plate. And this is due to first forming these small little vesicles containing some cell wall material inside of it. And these vesicles will then end up fusing with each other, forming a cellular plate like over here. Eventually, it'll fuse with the existing cell wall, forming this new cell wall here. And then you have two complete plant cells now. Good? Okay, let's see. Maybe it's a good time. Wait, is it a good time to stop right now? What time is it? Okay, we still have time. Okay, it's the cell cycle control system. This is when there's these proteins that actually help regulate or control the cell cycling. So they actually cause the cell cycle to stop at certain places to check for certain things. Some of them is just to check whether or not the DNA was replicated correctly. If it's not replicated correctly, it's going to stop the cell from continuing on. Because if you think about it, if DNA is not replicated correctly and there's mutations inside of it, do we want to undergo mitosis and cause all our future cells after that all have that mutation? No, right? So it's going to cause it to stop and cause it to first correct it first before going on. Other times, such as that are like neurons in our muscle cells, we want it to stop growing because it's at its mature process and it goes into its G0 state or resting state. Basically, it doesn't divide any longer. So most of our neurons don't divide any longer. The only ones that do divide is like our olfactory neurons, where for smell. And the reason why I had that cell control system involved is because cancer, they don't normally respond to that cell's control system. So if they don't respond to it, then we're having mitosis occurring without any kind of regulation. So mitosis will continually occur onwards and onwards. And then the thing is, most of the time the immune system will try to prevent that from occurring by destroying those cells, but it does miss it at certain times. And if it does miss it, that's where tumors starts to form. Okay? The tumors forms are these abnormal masses of cells that forms that lump, as shown over here. And benign tumors are when you have those cells, that mass of cells, but it just remains in that location, so it doesn't spread anywhere. That's a benign tumor. Go ahead, Paris.
Why do some tumors like develop like teeth and stuff?
Teeth? Oh, it does occur. It all depends on the tumor cell itself, the first tumor cell, whether or not it's been differentiated, if it's already programmed to form one thing or another. If the DNA was programmed to form teeth, it will form it into a tooth, kind of thing. Yeah, it does occur. However, benign tumors can still cause some sort of effect by causing neighboring organs to be compressed. If you think about a benign tumor in the brain, can cause a lot of problems because it might squeeze parts of the brain, causing damage to the neighboring neurons of the brain. Malignant tumor, on the other hand, is when they start spreading to neighboring tissues. And the spreading of neighboring tissues will cause, of course, damage to it. And also, it will help, might end up spreading through the blood vessels and lymphatic vessels too. And this is when we're actually called, this is called cancer at this point. Yeah. And then metastasis is when they actually end up going through the blood vessels and lymphatic vessels and start spreading to other organs, other locations in the body. Some treatments, surgery, of course, to remove the tumor. Which you're doing surgery, right? Taking out the radiation therapy is also another way of treatment. So you end up exposing that site to radiation. So hopefully you cause more harm to the cancer cells than the normal cells. However, of course, normal cells will also get damaged through the radiation. Chemotherapy, this is when you actually start using drugs and chemicals to help interfere with the cell division. Sometimes they stop the spindle fibers from producing. Usually, you do use this when there's metastasis happening. There's also ways to prevent cancer. First of all, don't smoke, right? Another thing is to exercise, decrease the amount of sun exposure, high fiber and low fat diet, self-examination, so to examine yourself whether or not you have certain tumors or abnormal lumps. And also regular visits to the doctors as well will help prevent cancers.
Would you mind going back to the radiation therapy?
Oh, it's just how radiation will help hopefully cause more damage to the cancer cells than the normal cells, but normal cells will also get hurt by the radiation. Okay, some terminology about sexual reproduction. Somatic cells is the reverse, it's the opposite of a gamete cell. Okay, so this is when we're talking about our body cells. So when we're talking about our body cells, how much chromosomes are in there then? 46, 23 pairs. So they are diploid. They're diploid, or 2N, right? So you always get one side set from your father's side and the other set from your mother's side. So that's where we get the pairs part, right? 23 from our father's side, 23 from our mother's side. Gametes, on the other hand, these are our sex cells, and these only contain 23 chromosomes in humans, and they're known as haploid, right? So just a single N. Females, they have eggs. Males, they have sperm. Fertilization occurs when you have the fusion of the egg and sperm together, and you produce what? A zygote, which eventually becomes an embryo, a zygote. Okay, and yeah, all those are sperm around the egg, but only one. Yeah, normally speaking, there are times that more than one occurs, but then they don't form a proper zygote. It usually dies off. It's when you undergo mitosis a little too many times, kind of thing. So that initial mitosis. Those are identical. Paternal twins is when you fertilize two separate eggs. Okay, so zygote, they contain how many chromosomes then? 23 pairs, thank you. Which is 46, okay. I thought you said 26. I was all like, where did you get that? Okay, 46, yes. 46 chromosomes, so they are, once again, 2N, which is diploid. Diploid, diploid. Tomato, tomato. Okay, karyotypes. Karyotypes is when you actually look at the appearance of these chromosomes and the amount of chromosomes that they are, and you match them up. These are then homologous chromosomes, meaning that the homologous chromosomes, these are when we were talking about the dad's side and the mom's side. We have one set here from our mom's side, one set here from our dad's side. And these are homologous, meaning these are the chromosomes that carry the exact same genes, same types of genes. They don't, genetically, they might not be the same, but they carry the same types of genes. So if we're talking about eye color, you'll find that in this particular chromosome at this location, kind of thing. So we have our sex chromosomes over here. You do see it over here. And you'll notice that the Y chromosome is a lot smaller than X chromosome. And the sex chromosomes is the one that determines the person's sex, usually by the Y chromosomes. That if you have a Y chromosome, you're going to become a male. Why?
Because they're tired from all that swimming around and keeping egg and stuff.
It's basically just it just gives enough genetic information to produce testosterone, so it doesn't need that much. So the default is to be a female kind of thing, unless you have a Y chromosome. Which it could be a good thing too, especially when you're talking about if you're missing chromosomes and stuff like that, like color blindness, like a lot of X-linked traits, sex-linked traits, disorders and stuff like that, like certain color blindness, or there are certain color blindness that are sex-linked. So for males, they tend to have more people with color blindness because their Y chromosomes don't carry the dominant genes or alleles for it, while females, they tend not to have color blindness because their X chromosomes that are on the other X chromosomes are the dominant alleles that will replace the sex-linked alleles that we were talking about that causes color blindness.
So many names for all those colors and stuff.
For females, they have 2 X chromosomes. For males, they have an X and the other chromosome is a Y chromosome. And the Y, as you can see, is a lot smaller than the X. Yes, the males determine the sex. Because the females, they have 2 X chromosomes, they won't be able to dish out a Y chromosome for their offspring. Autosomes, on the other hand, these are all these other chromosomes. So everything but the sex chromosome. Okay, all the other 22 chromosomes, 22 pairs of chromosomes. Okay, let's stop here and we'll continue on either later today or next time. I don't know. Maybe.
Let's just keep going.
How much? I know, seriously. You guys are hardcore. Yeah, you guys have youth with you guys. Okay. Meiosis. Okay. Meiosis is different from mitosis because we're actually forming gametes. And once again, gametes has 23 chromosomes. Yeah, gametes, right? Our sex cells, our sperm and egg. It makes sense, right? Why does our sperm and egg have 23 chromosomes?
Together they make 46.
Yes, a sperm 23 plus an egg 23 when they form a fertilized egg. Sometimes. Yes, there are some genetic defects of which we will talk about later, but if it happens normally, 23 plus 23 equals 46 chromosomes. Yeah. Alright, so meiosis is a cellular division to help us form the sperm and eggs, the gametes. So if we end up forming haploid cells, so only 23 chromosomes for humans, and it undergoes 2 cellular divisions, meiosis 1 and meiosis 2. And we'll talk about how it causes genetic variation as well. But we've already touched upon it a couple times through this lecture and also our last time we met up. Due to crossing over in prophase 1, and also random assortment, also called independent assortment, and metaphase 1 or anaphase 1, depending on how you interpret it. And we'll go into more detail as to what's happening there. Good? Okay. So, interphase. What's G1 again? Just think about it growing, right? Yeah, it's a gap phase to cause it to grow. So that it increase the amount of organelles, so that when it divides into 2 cells, it'll have enough organelles and stuff. At this point in time, the centrals also replicate. The S phase, that one should be easy, right? Synthesis, meaning that synthesizing what? DNA, for DNA replication. Very good. And G2, more growth happening. At this point in time, this chromatin condensing begins. I would say let's save that more for prophase, okay? But Basically, in G2, once again, it's growing as well in preparation of cell division. So in prophase 1, very similar. The nucleus dissipates, the nucleolus disappears. What else happens in prophase in mitosis? The chromosomes, not chromosomes, but centrioles go to opposing poles. But also, what you guys were saying, chromatin becomes, it becomes two sister chromatids become what? Alright, you're right. Chromatin is the diffuse form of DNA. When it condenses, it becomes a chromosome. Chromosomes, which is formed of two sister chromatids. Very good. Okay, and the two chromatids are joined together by the centromeres. Okay, so we've talked about the centrioles moving to the poles too. Very good. So all this is very similar, right? All this is exactly the same as prophase when we were talking about it in mitosis. The homologous pairs, the one from our mom's side here, and the one from I'll just keep it with color, our dad's side, and the one from our mom's side, they pair up, okay? At this point in time, crossing over occurs. And crossing over is where they end up having locations of the chromatids swapping with the homologous chromosome. So if I was to redraw it here, so that's one chromosome. Let's say that's from our mom's side, okay? And here's another one that I'm going to draw pretty much on top of it, okay? I'm going to just draw it a little thinner, okay? More or less. So they're kind of like right near each other, okay? I drew it kind of on top of each other. And what happens is that they start swapping parts of their locations on there. Basically, they're swapping genes here, back and forth. So then at the end, instead of having just this here and this here as their homologous chromosomes, because they swap parts of it with each other, they're going to end up having parts of it that's going to be from our maternal side and parts of it from our paternal side. So it's going to look more like, I'm going to draw this as being red over here. Let's draw this as being red over here and some over here as well. And the same thing happens. So basically they swapped parts of their chromosomes on either side. Does that make sense? They got so close together that they started swapping parts of the sister chromatids with one another. And you see that happening over here as well. That's crossing over. And the reason why this causes variation is because now we have something different, right, from what we started off with, right? Genetically speaking, right, we don't have what our mom's chromosome has exactly. We don't have exactly the same as our father's chromosome. We have like a hybrid between the two now, right? A little bit of each. Metaphase 1 is when the chromosomes line up again at the metaphase plate. So very similar. The spindle fibers attaches to the proteins next to the centrosome. And at this point in time, this is where some textbooks will say this is where the independent assortment or random assortment occurs. Other textbooks say that it occurs in anaphase 1. But anaphase 1 is when the spindle fibers starts to pull from the centrosomes, and they start to pull the homologous chromosomes away from each other. Okay, so now these homologous chromosomes are getting pulled away. And this is where that random assortment, independent assortment occurs, where it's randomly getting pulled away, meaning that if we're saying that this, and I'm going to draw another one here. Okay, and let me draw centrioles with this centrosome. Okay, so like these centrioles, this centriole here can pull this chromosome and this chromosome towards this side, and this centriole here will pull towards this side, right? So in the end, you have one cell that will have this chromosome pair, and the other cell having this chromosome pair. So far, so good, right? However, I could also say that this can pull this one here, while this one pulls that one. This one pulls this one pulls this one here. And now we have a different combination, right? Instead of having all blue on one side, we now have blue and red. Yeah, but it's a different combination, right? So this is creating more genetic variation occurring. So we want to have more genetic variation in case some environmental change occurs. Someone has certain traits that are more beneficial for that changed environment. Okay. Once again, at the end of anaphase 1, beginning of telophase 1, cytokinesis begins to cause that cleavage furrow. Telophase 1, exact opposite of prophase 1, where the chromosome starts to decondense again. The nuclear membrane will do what? Start to reform. Very good. And cytokinesis will finally complete. And the nucleolus reappears as well. So it's basically the exact opposite. At this point in time, after cytokinesis finishes, how many chromosomes are there now? Like at the end of this process? Okay, originally we had 23 pairs, right? After we undergo DNA replication, right, it's 23 pairs times 2, right? Okay. That's when we began, right? This process, right? 92-ish chromosome. 92. Okay? And then, after what happened in anaphase, where you pulled one homologous chromosomes to one side and the other homologous to another side. Okay? So let's talk about it. Originally, it's 23 pairs. When we're talking about pairs, we're saying one set is from our mother, the other set is from our father, right? And then when we replicated it, it's just 23 pairs times 2, right? During anaphase 1, we're taking one set or the other, okay? So it's not going to be paired off anymore, okay? We're going to just be, so anaphase 1, we're only going to be taking one set or another, right? So there's going to be 23, basically times 2, right, still? And then 23 times 2. Notice how it's not a pair anymore. So is this a haploid cell or a diploid cell? Haploid, right? Because we don't have that pairing anymore. We're either going to have one set, if the meiosis 1 occurred just taking all the mother side, just to keep it simple for now. So this is going to be only 23 all from our dad's side, and this is 23 all from our mom's side, okay? So it's not diploid anymore. If it's diploid, it will still have that homologous chromosome together, where you have one set of mom and one set of dad, okay? In actuality, you don't get all one set from your dad's side or all one set of mom's side. You're going to get, since it's randomly assorted, right? You're going to have some that are from mom, some from our dad, but then vice versa on the other side. Okay, this times 2 we'll talk about later, but after meiosis 2, that's when you divide it again and only have 23 chromosomes on either side. So you'll end up having 4 haploid cells. Okay, so at the end of meiosis 1, we have 2 haploid cells, which is what we have here. Okay, prophase 2. You guys could tell me this. What happens in prophase? The disappears. Nucleolus and nucleus disappear. What else happens? The chromosomes condense. Very good. The centriole moves to the poles. Yeah, very good. Okay, and there's spindle fibers that will try to attach to the centrosomes of the chromosomes. Metaphase 2, what's going to occur? They're going to line up at the chromosomes line up at the metaphase plate. Very good. Spindle fibers attached to the centromeres. Anaphase 2. Now this is where the centromeres actually break now. Once they break, the sister chromatids will be pulled apart, becoming daughter chromosomes. Yeah. So now they go to their opposing poles. Cytokinesis begins to cause that cleavage furrow. Oh, sorry. But it's basically the same thing as what we've... This is... Meiosis 2 is almost identical to mitosis. Finally, in telophase 2, what's occurring? You guys could tell me this. Yeah, what condenses? The chromosomes. Is it condensing or... Decondensing, yeah. The chromosomes decondense. What else happens? The nucleus, what? Form again. Yes, the nucleus membrane forms again. Nucleolus. It comes back as well. Very good. The centrioles dissipates their spindle fibers. And finally, we finish cytokinesis occurring. And at the very end of this, we have 4 haploid cells. Yes, there's 2 phases. There's meiosis 1 that occurs, and then meiosis 2. And notice between meiosis 1 and meiosis 2, was there interphase? No, there was no interphase. So we don't replicate DNA again, and it just goes straight to meiosis 2. Okay, so we already a little touched upon all the different ways of genetic variation. We talked about crossing over. When does it occur again, crossing over? That's cytokinesis. Yeah, during prophase 1, that's when you end up having crossing over. And once again, this is where you have the homologous chromosomes. They start swapping parts of themselves with each other. Chiasma is the location where the crossing over actually occurs. Independent or random assortment, when does this occur? During metaphase 1 or 2? 1. Yeah, metaphase 1. Some places they say anaphase 1, depending on how you look at it. But notice how both of these genetic variation occurs in meiosis 1. Okay. So at this point in time, the homologous chromosomes, they align at the metaphase plate, and then they are randomly chosen as to going to which pole it will end up going to. So that's why it's called random assortment or independent assortment, is because it's randomized in which cell it will eventually go to. So once again, this is one possibility is if you end up only having two pairs of chromosomes, total of 4 chromosomes as this cell did. Random assortment will cause these two haploid cells to form. Or if you were to choose this set here, or this set, notice how you eventually get different types of cells, right? The different types of chromosomes in the cell. You could get all blue or all red, or you could end up getting one blue and one red, and then the other is the vice versa of it. So notice how at the very end, our gametes here have different types of chromosomes inside of it. There's the way to calculate the number of different types of combinations available is seeing how many cells you end up forming, which is 2 after this part here. To the power of n, and the n is the number of haploid chromosomes you have. So in humans, they have how many? 23, yes, So it would be 2 to the 23rd, which is a huge number, over 8 million strong. So this is the different types of combinations that can possibly occur. Random fertilization is yet another way of genetic variation. Since we're going to end up having different types of sperm and egg develop, those combination of those 2 sperm and eggs fusing together will have different type of combinations as well. So of course you're going to end up multiplying it to the square or multiplying it by itself. So you end up having this many different types of combinations available. You don't have to memorize the numbers. It's just showing you the how unique we are, how many variations of the different types of gametes that we can form and then fuse together to form 1 out of 70 trillion will be identical with each other. And even that's not considering the crossing over portion of it, right? We're just talking about independent assortment and this random fertilization occurring.
So when it comes like identical twins, are they off by a certain?
No, identical twins were actually undergoing mitosis from after it's fertilized.
Yeah.
So after it's fertilized, so it's not. We're not talking about 1 out of this many to have the same genetic material. This is like if you're saying like you have a child, right?
Yeah.
And then your second child to have the exact same genes. Your second child is 1 out of 70 trillion. It is possible, but very unlikely, because when we're talking about unless there's no crossing over or an exact same crossing over occurs, which is even more rare, so you're going to increase the number about that. So, however, there are some mitotic mistakes that can occur, and this is called non-disjunction. When chromosomes, when they pair up, they don't separate completely. So then you end up having a different amount of chromosomes at the end. Notice over here, meiosis 1 did not end up dividing correctly with the amount of chromosomes. So this cell has more chromosomes, this has less chromosomes. So the gametes here, there's a lot more and a lot less over here. This is yet another example here where you end up having more chromosomes that occurred in meiosis 2, not in meiosis 1, but it can also occur as well. These are normal here, right? These here are normal. This is abnormal. So if fertilization occurs, with one of these abnormal amounts of chromosomes, then you're going to end up having a zygote with abnormal amounts of chromosomes as well. And this zygote will undergo mitosis, and so all its cells will have this abnormal amount of chromosomes as well. The example is trisomy 21. Another one is the abnormal amounts of sex chromosomes. Most case scenarios, these fertilized cells die off. Okay, so they're not going to produce a functioning human at the end. However, trisomy 21, we do actually have that occurring. Trisomy 21, like the name implies, there's 3 chromosomes of chromosome number 21, as shown over here. There's 3 of them. So this is someone with Down syndrome. It does occur 1 out of 700 births. Characteristics, they tend to have a round face, flattened nose, tends to have short stature, some heart defects, and depending on different degrees of mental difficulties that they might have. As you can see from this chart, as the years of the mother increases, the number of births with trisomy 21 increases as well. As you get older, as women get older, the more chances of having a trisomy 21 to occur. Exactly. Yeah, they do tests for mothers who are above a certain age. Oh, it's chromosome number 21 over here. So it's not male or female kind of thing. It's just one of the autosomes chromosomes, not the sex chromosomes. However, talking about sex chromosomes, Klinefelter syndrome is when the male actually has an extra X chromosome. They're still male because they have the Y chromosome. However, yeah, they have more feminine features. So they also tend to have small testes and are sterile. They have enlarged breasts, and also tends to have a feminine type of body shape. Yeah, You can't really tell because, well, I mean, if I had a side view of it, I guess you could kind of tell a little bit about the enlarged breast a little bit. But it's not, I guess you could kind of say that he looks like an overweight person too, potentially. The thing about people with fat tissue, they produce a lot of estrogen as well. So that also comes into play when we're talking about people who are a little larger for males. Males with an extra Y chromosome tend to be just taller. It used to be when I was back in the day when I was learning biology, the Y chromosomes, if you have like XYY or XYYYYY kind of thing, they said that they tend to be people who are more aggressive or people who tend to be aggressive to the point that they end up getting locked up in jail and stuff. However, I think that's kind of proven to be incorrect. So all it is that these males tend to be a little taller. A little taller, yeah. I don't know how many inches or anything. They just tend to be taller in general. Go ahead.
So this is different from a hermaphrodite?
No, different from a hermaphrodite because they still have their genitalia. However, they don't have both genitalia. They only have these two have male genitalia. It's the secondary features for the Klinefelter. Secondary feature meaning not their genitalia, but the breast size and stuff like that tends to be more feminine.
But you said that they're sterile, so
they can't reproduce. Yeah, meaning that their sperm are not viable to fertilize an egg.
So it's not just their secondary, but maybe their primary.
What I'm trying to say is that they don't form female genitalia. Okay.
These two different, right?
No, both of them will have male genitalia. They both have testes and a penis and stuff like that. However, the one with the Klinefelter syndrome will have smaller testes and have female secondary characteristics like enlarged breasts kind of thing.
The hermaphrodite has a female reproductive.
Yes, hermaphrodite will have both female and male reproductive organs kind of thing. See that functional? Yeah, That may. Now that more I talk about, it, I think usually it's one or the other that has functioning. I don't think they're. I think they don't necessarily have to be sterile.
So do they take the one that's not functioning away?
That's up to the parents to decide and the individual to decide, depending on at what point in time they had that decision, kind of thing. Females with an extra X chromosome. Okay, so give me an example of a male with an extra Y chromosome. I mean, sorry, not that kind of. I mean, like, give me an example as for their sex chromosome. Like, so Klinefelter syndrome is when you have an extra X chromosome, and you have this kind of sex chromosome here. So males with an extra Y chromosome, XYY, yeah. Okay. Females with an extra X chromosomes are hard to differentiate with normal female. Okay, it doesn't make them extra sexy by having an XXXX chromosome. They actually have very similar characteristics as a normal female. It's just a normal female. It's just, yeah. Yeah, the only way you could tell is by seeing the karyotype. If you remember the karyotype, the thing that we saw here, this karyotype, over here on the X chromosome here, there will be a third one there, kind of thing. Okay. Turner syndrome. This is when a female has only one X chromosome. It doesn't have that second X chromosome. So they usually call it XO. These females tend to be shorter. They tend to have this web towards a web of skin around their neck area, as this individual is shown here. Yeah, They did take it off, and so she doesn't have that as much. These females tend to be sterile as well, so won't be able to reproduce. I don't know how common. I don't think it's that common though. They also have a poor development of breasts and also secondary sexual characteristics, so they tend to look more young. Even though they can age, they tend to have more young. They don't develop those things, so they don't. They look like as if they didn't go through puberty, type of thing. Okay, so we're done. So we went through that pretty quick, I